I've ever met. He's incredibly helpful to people in the lab. Anyone who asks him to do things, he will. I've asked him to do countless things. Sorry, end of a cold. I probably had a mentor, you know, it probably seems like 100 students to him, from undergraduates to rotation students to new people in the lab, and he's done it, you know, without, you know, unbegrudgingly and with, you know, with a real pleasure. He's also a very hard worker, and I think that work ethic has really held him in good stead. I think he realizes how hard it is really to not only complete the work for a thesis, but actually produce the document. It takes a while, and I think you know, because of his hard work and his diligence to the project, it really, I think, turned into a great story. Another thing about Mark is he's you know, for his age, he's still very young. He's incredibly mature. And I think he's been buoyed by both his family and his faith, which he holds dear. And I just want to thank him for joining my lab. He's been a great part of the lab, and I'm happy he's going to stay on for a little bit longer, and for all the hard work that he's done. So with that, I want to introduce the talk today. He's going to give a talk that's the title of which is Crosstalk between brain-derived neurotrophic factor and glucocorticoid receptor signaling.
Hi, thank you all for coming. Thank you, Michael, for that kind of introduction and for the opportunity to work on this project in your lab. So I'll give you a brief introduction about what I'm going to talk about. It's going to be about glucocorticoids, what they are, and what is BDNF, brain-derived neurotrophic factor. And then I'll tell you a little bit about the aims of my project, the questions I intend to address, and then the rest of my presentation is broken up into 3 parts. Part 1 focuses on how BDNF specifies and amplifies the GR transcriptome in neurons. Part 2 really looks at how BDNF is able to induce GR phosphorylation, and we look at how that modulates GR target gene expression. And then Part 3 focuses on the effect of GR phosphorylation to physiology. Specifically, I'll show you data about neurosynaptic plasticity and GR phosphorylation's role in that. So glucocorticoids, they are primarily used therapeutically as an anti-inflammatory and immunosuppressive agents. They're secreted from the adrenal cortex of the adrenal gland, and they're secreted in a circadian manner or in response to stress that's perceived by the brain. Now they're able to modulate physiological homeostasis and coordinate those adaptive responses to stressors. And an imbalance in glucocorticoid levels, specifically chronic glucocorticoids, has been associated with a number of neuropsychiatric disorders, specifically depression, post-traumatic stress syndrome, and even bipolar disorders. So glucocorticoids can act through and activate the glucocorticoid receptor, or GR. GR is an intracellular steroid hormone receptor. In the absence of steroid, GR is typically localized to the cytoplasm of the cell, where it is bound to heat shock protein complexes. Upon steroid binding, or ligand binding, GR is able to dissociate from this complex and translocate to the nucleus, where often it homodimerizes at glucocorticoid response elements, or GREs. Now these specific response elements help to target GR target genes, where it's able to induce or repress transcription. So in a sense, it acts as a ligand-activated transcription factor. So there are several different types of GREs. One, you have simple, composite, and tethering. So simple GREs typically are composed of an inverted repeat sequence separated by 3 random nucleotides, and this supports GR homodimerization on DNA. Then you can have composite GREs that contain a binding site for another transcription factor other than GR. And sometimes this is able to coordinate activity to induce transcription. And then in tethering GREs, you typically have protein-protein interactions with another transcription factor. And this often, in this case of this GRE, is often not a typical consensus GR binding sequence that is mediated through protein-protein interactions. Now, You can also have negative GREs of these three types as well, but the majority of my talk will focus on transcriptional induction and the use of these positive GREs. So GR is expressed widely in the body. It's expressed also widely in the brain. Figure A shows you an in situ hybridization for rat GR mRNA. And you can see that GR is localized in regions of the cortex, the hippocampus, the amygdala, even parts of the hypothalamus. And by Western blot, we see total protein levels in various tissues of the brain, the cortex, hippocampus, and hypothalamus. And they're comparable to levels of GR within the liver and the lung. We've also, via immunofluorescence, have been able to find that GR is localized in neurons, because there's other types of glia or supporting cells in the brain. So GR is localized in neurons of the cortex and hippocampus, as you can see here. So, as I mentioned, we've been interested in how GR signaling and BDNF signaling inside neurons kind of cooperate to regulate different functions within the central nervous system. Now, BDNF, or brain-derived neurotrophic factor, is a peptide hormone. It binds to receptors on the membrane of cells known as tyrosine kinase receptors. But these receptors are specific for BDNF, and they're called TrkB receptors. And upon binding to BDNF, they dimerize and activate several downstream kinase signaling pathways, like MAP kinase, AKT, PKC. And these, in turn, activate transcription factors, some of which are CBPs, ELK1, CREB, and BDNF signaling has been very important for neuronal survival, differentiation, and just general growth of neurons. Now, GR also has functions within neurons that many actually share some of these functions with BDNF signaling. Of these are neuronal survival, neurotransmission, remodeling of axons and dendritic spines, and even synaptic plasticity. So there's a relationship between BDNF levels and GR levels in the brain, specifically in the hippocampus. A recent study from the Journal of Neuroscience shows that in GR heterozygous mice, you can have reduced levels of GR compared to wild-type mice. And this correlates with reduced levels of BDNF, specifically in the hippocampus. Now, if you look at the levels of another protein, NGF, you'll find that these levels are not sensitive to reduced levels of GR, suggesting some type of specificity between the BDNF levels and GR, that crosstalk. Now, if you overexpress GR using a yeast artificial chromosome, you can have higher levels of glucocorticoid receptor protein, and you also, that can be correlated with higher levels of BDNF in the hippocampus. And again, this is specific for BDNF as opposed to NGF. Now, these mice have been shown to have a depressive-like phenotype, while the overexpressing GR mice are resistant to stress. So the questions that I intended to address in my thesis work, really, what is the impact of BDNF signaling on GR function in neurons? Because there's a lot that we don't really understand about the molecular mechanisms of this crosstalk. And more specifically, is GR transcriptional regulation affected at all by BDNF treatment? So part one of my talk, I'll demonstrate how BDNF is a modulator of GR-induced transcription. So to assess the direct effects of GR transcriptional activity from BDNF treatment, we engineered HEC293 cells to stably express the TRK receptor, which is the receptor for BDNF. And we've also transfected in GR. Now, this system allows us to also transfect in another reporter gene, TAT3 luciferase, which contains several simple GREs, or 3, upstream of a promoter that drives luciferase expression. So if you treat these cells with dexamethasone, which is a synthetic glucocorticoid, you should see higher levels of luciferase, which correlates with GR transcriptional activity. And that's exactly what we see. The cells treated with dexamethasone, we see an induction of GR transcriptional activity, while treatment with BDNF really has no effect. Now, we've also shown, I don't have the data, but I'll tell you that treatment of these cells with BDNF does not induce the translocation of GR from the cytoplasm to the nucleus. So this was kind of an expected effect. Now, upon co-treatment of BDNF and dexamethasone, you now see enhanced or amplified activity compared to dexamethasone alone. And this is not due to any changes in protein levels, since the protein levels generally stay the same on each of the treatments. We know this is an effect that's dependent upon TrkB, which is the receptor for BDNF, since in 293 cells that do not contain the Trk receptor, we do not have this enhanced activity upon co-treatment. Now, we were interested if these effects could be noticed at an endogenous GR target gene. So we took two genes. One is SGK1, which is a kinase that's involved in regulating sodium and potassium and chloride channels, even in neurons, and GILS, which encodes for a transcription factor that regulates apoptosis. So if you treat with dexamethasone, just like the TAT3 reporter, you see induction, while BDNF has minimal effect. But upon co-treatment, you now see this enhanced activity, very much like what we saw with the TAT3 reporter. But GILS, on the other hand, GILS, upon the addition of BDNF, did not enhance activity compared to just dexamethasone alone, suggesting that the effect that BDNF has on GR transcriptional activity seems to be specific for select genes. Now, we were interested whether BDNF could affect the recruitment of GR to the promoter or to DNA at these genes. So we designed primers around the GRE within the SGK1 proximal promoter and primers around the GREs within GILS. And then we performed chromatin immunoprecipitation experiments upon dexamethasone and BDNF plus dex treatment, since BDNF alone did not affect translocation. So upon dex treatment, we see what we would have expected, is that you have increased GR recruitment to the SGK1 promoter, but then now upon BDNF and dexamethasone co-treatment, you now see this enhanced recruitment, a significant enhanced recruitment of GR to the SGK1 promoter. Now, we also did the same experiment for the GILS GREs. We noticed that upon dexamethasone treatment, you actually have a little bit higher GR recruitment, but now upon co-treatment, you also tend to increase this slightly, but it's not significant. So this suggested that BDNF treatment was able to increase GR recruitment to the SGK1 promoter and slightly at the GILS promoter, suggesting that this may have an effect on GR transcriptional activity, specifically the enhancement that we see at SGK1. So we wanted to know, in primary neurons, are there selective genes that recapitulate the effect that we see in the 293 TrkB cell system? So we took primary rat cortical neurons, and I've shown you before that GR is expressed in the cortex, and it co-localizes with TrkB, the receptor for BDNF, as you can see here. So we cultured primary rat cortical neurons, treated them with either dexamethasone, the synthetic glucocorticoid, BDNF, or BDNF plus dex. We extracted RNA and performed microarray profiling to determine those genes that were sensitive to specifically co-treatment and dex and BDNF alone. So what we find is that there are about 450 genes that are responsive to dexamethasone treatment, while the repressed genes are indicated by green and induced by red. We also find a number, about 450 genes that are also responsive to BDNF treatment. Then, when we co-treated, that seemed to double that amount. About 900 genes were responsive to both BDNF and dexamethasone co-treatment. Now, when I first analyzed this data, it really didn't seem surprising that it was because you have dexamethasone, which seems to induce one signaling pathway in BDNF, which can affect another signaling pathway, that now upon co-treatment you have the induction of both of these pathways. And that would probably be represented with a Venn diagram something like this, where the dex transcriptome can overlap a little bit with BDNF, but now upon co-treatment it encompasses both signaling of dexamethasone and BDNF. But in actuality, when we analyzed the crosstalk between the genes, we found that majority of the co-treated genes were uniquely responsive upon co-treatment. Nearly 50% of the genes were responsive only to co-treatment, meaning you treat with BDNF, you treat with DEX, they were not responsive above twofold, but now if you treat with co-treatment, they were now responsive above twofold. And when we analyzed many of these genes by gene ontology analysis, we found that they were correlated with synaptic transmission, synaptic plasticity, axon guidance, many of the functions that GR has already been demonstrated to modulate in neurons. Then we also have a class of genes within the DEX responsive circle that were induced by dexamethasone and then enhanced above twofold by BDNF. And that represented about 30% of DEX responsive genes. And this, we thought, was very similar to what we saw with the TAT3 promoter, where you can have induction by dexamethasone and then enhancement or amplification by co-treatment. So we validated a number of genes from our microarray using real-time qPCR. This is represented by the heat map that you see here, with red indicating higher expression and green lower expression or repression. So we validated, we chose many of the highly expressed genes by co-treatment and some of the repressed genes by co-treatment. And you'll notice that many of these genes are induced by dexamethasone. Some are not even responsive to BDNF, but now enhanced by co-treatment, very similar to what we saw before. And then even some of the repressed genes show a similar profile, repressed by dexamethasone, and even further repressed by co-treatment. So what you could do, you could classify many of these genes into several different types, depending on how they respond to co-treatment. So you have some genes that are responsive to DEX and enhanced upon co-treatment. Some genes that are induced by DEX, but not really affected by the addition of BDNF. And then you have other genes which are induced by DEX, and actually the induction is repressed or not as amplified upon co-treatment. Now, if you take a closer look at this type 1A, you can even separate this further, where you have some genes that are even responsive to BDNF. Here, there's not much of a response to BDNF, and then you have amplification. Here you have responsive to both of the individual treatments, and then you have what seems to be a synergistic response upon co-treatment. Now, the majority of DEX responsive genes were of the type 2 class. Only 30% were of type 1. But we still thought that this represented an interesting phenomenon, an interesting crosstalk between BDNF and dexamethasone signaling. So the rest of my talk really focuses on these type 1 and trying to understand a potential mechanism behind that. So the last thing we did with the arrays, we wanted to determine whether our genes, our validated genes, were primary GR target genes. So presumably if a gene was a secondary target, you could in some cases have GR activating or inducing the RNA expression of a primary gene, which would be translated into protein A, which would modulate the secondary gene expression. Now, if you pre-treat with the drug cyclohexamide, which blocks protein translation, you would see a different effect upon cyclohexamide pre-treatment. So we analyzed our genes. The wild-type conditions are without cyclohexamide pre-treatment, and then pre-treated those genes with cyclohexamide, and 10 out of the 13 genes responded with very similar profiles with or without cyclohexamide, suggesting that the majority of the genes are primary GR target genes. So to summarize, BDNF via TrkB increases GR transcriptional activity of a transfected reporter gene, suggesting a direct effect of BDNF signaling on GR transcriptional activity. And GR recruitment seems to be also enhanced at a GR target gene, SGK1. Now, this surprised us as well. BDNF and glucocorticoid co-treatment seems to induce a unique set of genes in neurons, represented by the 48% that I showed you in the Venn diagram. BDNF treatment also amplifies many GR target genes in neurons, and this is despite minimal induction upon BDNF treatment alone. And 10 out of the 13 genes that we tested that are DEX responsive do not require new protein synthesis to induce gene expression by GR. Therefore, we classifier considered those to be primary GR target genes. So part 2 of my talk will focus on the role of BDNF-induced GR phosphorylation. So glucocorticoid receptor is a phosphoprotein. It can be induced, phosphorylation can be induced by several upstream kinases, including MAP kinase signaling, CDKs, and GSK3 beta. And we know that this can have an effect on protein stability, or GR turnover, subcellular localization, recruitment to DNA, and especially cofactor or coactivator interactions. And we believe that this can have a specific effect on gene expression. You can have essentially a gene expression profile depending on the phosphorylation status of GR. Now, BDNF and tract B signaling is able to induce MAP kinases and can indirectly affect CDKs. I think this has an effect on GR phosphorylation, which I'll show you in my talk, that affects cofactor interactions, and I believe that this will generate a specific gene expression profile that's different from dexamethasone treatment alone. So my hypothesis for my thesis entering this project was that BDNF signaling is able to induce GR phosphorylation, which results in the enhancement of GR transcriptional activity. And I'll go ahead and show you a model for how we think this occurs. So in the absence of BDNF or tract B signaling, we find that upon glucocorticoid treatment or dexamethasone treatment would lead to the phosphorylation and activation of GR, which would induce select GR target genes. Now, upon tract B or BDNF treatment and tract B activation, you can have the induction or activation of downstream transcription factors at certain GR target genes. We think that is important because also tract B is able to induce GR phosphorylation, and together they're able to recruit a third coactivator, which we think leads to the enhanced transcriptional activity that you see, that's shown you before. So, by mass spectrometry in collaboration with Tamer Nubis' lab here at NYU, we were able to discover 2 phosphorylation sites on GR that were induced by neurotrophin or BDNF treatment in neurons. So we used our 293 tract B system to just characterize these sites and specifically phosphorylation-specific antibodies to these sites. The sites are serine 155 and serine 287, and they're located within the N-terminal domain of GR, along with several other sites that have been shown to be important for GR transcriptional activity upon glucocorticoid treatment. These sites, 155 and serine 287, are conserved in the rat, mouse, and human, and using 293 cells that express wild-type GR or a phosphorylation alanine mutation at the sites, we were able to characterize phosphorylation-specific antibodies, since BDNF treatment in the wild-type cells induces serine 155 phosphorylation, but in the mutated cells, we don't really see any immunoreactivity despite induction or activation of tract B. Now, we see the same thing for serine 287. BDNF is able to induce phosphorylation at this site, which we don't see with the mutated GR, which mimics phosphorylation inhibition. Now, I told you before that there are several other phosphorylation sites within the internal domain that we've shown some of these sites can have an effect on GR transcriptional activity. So we wanted to know, does BDNF affect these other sites as well? And essentially, BDNF is able to minimally induce these sites, but the greatest induction is by 155, serine 155 phosphorylation, and serine 287 phosphorylation. Dexamethasone is able to induce many of the other sites, but it also seems to have an effect on serine 287, which is one of the sites we identified in the mass spec. So we were curious, if BDNF is able to induce serine 287 phosphorylation and dexamethasone also has a response for this site, what about co-treatment? What would be the response upon co-treatment? So we find that upon co-treatment of the 293 tract B cells, serine 287 phosphorylation is enhanced, And when you quantify that with total GR levels, you find that there's at least nearly a 5.5-fold induction. So we also can validate that these sites are phosphorylated in actual primary neurons. Upon BDNF treatment, you see sensitivity or immunoreactivity from both the 155 and serine 287, but the serine 155 is not sensitive to dexamethasone treatment, while the serine 287 is after even just 30 minutes, and co-treatment will induce both of these phosphorylation sites. So we were curious about the kinetics of 155 and serine 287 induction. Is it a rapid response? Does it take longer than 1 hour? So we used our 293 type B cells and treated them with BDNF from at least 5 minutes on up to 4 hours. We noticed that there was an induction even after 15 minutes, with most of a maximal response at both of the phosphorylation sites after 30 minutes of treatment, suggesting that the effect was rapid from BDNF signaling. We even took our 293 cells, we treated them with dexamethasone, and we don't see much of a response from serine 155, as it also I've shown you that it's not very sensitive to dex treatment, but serine 287 is sensitive. And about after 60 minutes of dexamethasone treatment, you see a maximal response of serine 287. So BDNF can have a rapid response for inducing these phosphorylation sites, and we think that that may be key for data that I'll show you later. So what we've come up with is a model for a GR phosphorylation code dependent upon the treatment. Dexamethasone is able to induce several GR phosphorylation sites, and we know that from previous work in our lab and other labs that this can have effect on coactivator recruitment. Now BDNF does not activate the receptor or induce translocation, but is able to induce phosphorylation at several sites, serine 155 and serine 287, more than any of the other sites. Now, if you co-treat with both BDNF and dexamethasone, you now have an activated receptor that's been translocated to the nucleus and rapid induction of serine 155 and serine 287. And we think that that has a specific profile or phosphorylation code that's different from dexamethasone. And we were interested in seeing what the effects of this phosphorylation code was on transcriptional activity. So to summarize briefly, serine 155 and serine 287 are GR phosphorylation sites that rapidly induced by BDNF treatment. 155 and serine 287 seem to show the greatest fold induction in response to BDNF compared to untreated cells. And serine 287 is responsive to both treatments and seems to be enhanced upon co-treatment, while serine 155 responds to BDNF but not dexamethasone treatment, so BDNF dependent site. And the co-treatment seems to, we would argue, create a unique phosphorylation profile. So in the next few experiments, I'd like to look at many of our GR target genes to see whether GR phosphorylation has an effect on mediating induction of these target genes. So we created an alanine double mutation at the two created sites, serine 155 and serine 287, and then we assessed by real-time qPCR transcriptional activation. So SGK1, I've shown you this before, it's enhanced upon co-treatment, and in 293 type B cells that stably express wild-type GR or the alanine double mutation, you'll notice that it seems that phosphorylation has an effect on mediating transcriptional induction of SGK1, since the double mutation has diminished induction. FKBP5 is another GR target gene that seems to be sensitive to the phosphorylation site, since the double mutation has diminished activity. Now GILS. GILS is a gene that's not enhanced upon co-treatment, and the phosphorylation sites also seem to not really affect its transcriptional induction. And CMYC is a repressed gene. Upon dexamethasone treatment, you see at least a twofold repression. Also, this gene seems to be phosphorylation insensitive. So we went to our primary neurons and we looked at many of the genes that we identified in the microarray experiment, and we found that NGFIB, PRL3C1, SRGN, all of these genes seems to be sensitive to the phosphorylation at serine 155 and serine 287, since upon co-treatment you see diminished induction. But myomason 2. Myomason 2 seemed to be a special case. Upon co-treatment, you see enhancement of this gene, but it was not sensitive to phosphorylation, suggesting that you had something that was specific about these genes that determined whether it was going to be phosphorylation sensitive or not. But it seems that for many genes, phosphorylation mediates the transcriptional induction. So we were curious, are phosphorylation sensitive genes distinguished in any way by the phospho-GR isoform recruitment to that gene? So for example, if you have GR that's highly phosphorylated at serine 287 and serine 155, does that go to SGK1 as opposed to GILS, a phosphorylation insensitive gene? So we tested this experiment by doing a chip chromatin immunoprecipitation using our phosphorylation specific antibodies, the serine 155 and serine 287. And what we found was that I've shown you the total GR chip experiment before at SGK1, that dexamethasone is able to increase GR recruitment, and co-treatment enhances that recruitment. Now, using the serine 155 phosphorylation antibody, upon dexamethasone treatment, we did not see much GR recruitment to the SGK1 promoter. But that correlates with the fact that serine 155 is not induced upon dexamethasone treatment. But now upon co-treatment, we now see GR recruitment to the promoter. Serine 287, on the other hand, is sensitive to both BDNF and dexamethasone. And upon dex treatment, we see induction of serine 287, or recruitment of GR at serine 287. And upon co-treatment, we see this phosphoisoform recruitment enhanced. Now, at GILS, we see a very similar profile, suggesting that the differential effects of GR phosphorylation on gene expression are not caused by differential recruitment of phospho-GR, since the phospho-GR isoforms appear to go both to phosphorylation-sensitive gene and a phosphorylation-insensitive gene. I actually presented this data to my colleague, and she said, "Oh, that's so sad." But it was kind of disappointing, but it actually made me think about post-recruitment effects. So in this model, we find that something about the gene, which would actually happen to be a transcription factor binding site, really helps to specify its phosphorylation sensitivity. And in our model, we find that BDNF and TrkB signaling is able to enhance transcription factor binding to certain GR target genes. And this, coupled with the phosphorylation, is able to recruit coactivators that we think mediates the transcriptional induction of GR select genes. So I'll take you back to SGK1, where we really took a biased approach. We performed chip experiments using antibodies to GR, and then several coactivators that we knew interacted with GR, and one of the downstream transcription factors of BDNF signaling. Now, it happened that a transcription factor binding site was very close to the GRE within the SGK1 promoter, so we looked at Krebs recruitment. So before I show you, we did this experiment just under two conditions, vehicle and BDNF plus dex. And we did this experiment using the stable wild-type GR expressing cells and the double mutant phosphorylation mutant expressing cells. And before I show you the data from the double mutant, I'll walk you through the expression of each of the coactivator recruitments to the GRE within SGK1. And what we find is that upon BDNF plus dex treatment, you have increased GR recruitment, which we'd expect. I've shown you before. Not much of a change upon GRIP1, minimal. BRG1, which is a chromatin remodeling protein, not much of a change. But P300, it's a histone acetyltransferase, seems to be recruited to this GRE region and enhanced upon co-treatment. CREB, downstream of BDNF signaling, also enhanced upon co-treatment. So what we find in the double mutant expressing cells is that GR is recruited to a very similar trend or a very similar pattern. Now this makes very much sense with the data that I showed you with the phosphoisoform recruitment. suggesting that the phosphorylation status does not dictate the recruitment of GR to the promoter. So it was not surprising to see this result. GRIP1 doesn't really have much of an effect, nor BRG1, but P300 was sensitive to GR phosphorylation since it was not enhanced upon BDNF plus dex treatment in these double mutant expressing cells. CREB also was sensitive to GR phosphorylation. So this told me or suggested that BDNF-induced GR phosphorylation was able to stabilize the recruitment of both a coactivator, P300, and a transcription factor, CREB, to the SGK1 promoter. So we wanted to know whether CREB was even involved in the transcriptional induction of SGK1 upon BDNF plus dex treatment. So we performed knockdown experiments using siRNA to CREB, and we've knocked down this transcription factor, or the levels of it. And then we assessed by real-time qPCR SGK1 expression in the 293 tract B cells. And we find that using the scramble, there's an induction very much like what we see before, but that induction is diminished in the cells expressing siCREB, or the knockdown of CREB expression. Now Gills is insensitive to phosphorylation. We find that it's induced upon BDNF plus dex treatment, but the induction is very similar upon the knockdown of CREB, suggesting CREB is not involved in the induction of Gills. So that brings us to a model in which BDNF and tract B signaling are able to do three things. One, it's able to activate downstream transcription factors at GR target genes. Two, it's able to phosphorylate GR at two sites more than others, serine 155 and serine 287. And three, it's able to increase or enhance the recruitment of GR to the promoter. And we think that these effects have an effect on coactivator recruitment, such as P300, which is mediating the transcriptional enhancement of select GR target genes. Now, if you mutate the phosphorylation site on GR, you diminish CREB recruitment and the coactivator P300, and you also diminish transcriptional induction. So this effect could be mediated by physical interactions, but that's something that we have yet to kind of solidify. So in the final half of more than half, in the final couple of slides, I'd like to tell you about a role of GR phosphorylation in dendritic spine number and morphology. This work was conducted by Dr. Freddie Jonatu in the Chow Lab. And essentially, you can count dendritic spines. So dendritic spines are really protrusions or sprouts that extend from dendrites of neurons. So here's the cell body of a neuron, and you can have dendrites that extend from that cell body, and on the dendrites are protrusions or spines that mediate interactions with other neurons within the brain. And these spines have been shown to be important because they serve as a storage site for synaptic strength. They bind to axons from other neurons. And they also serve to increase the number of possible contacts between neuronal axons. Now, spines, it is thought in the field that they can be actually an anatomical substrate for memory storage and synaptic strength because of their interaction with other neurons. And you'll find that in patients who have learning or memory impairments, that correlates with reduced spine number, or even some cases of depressed patients that correlates with reduced spine number. So you can culture neurons, and you can actually count the number of spines on a dendrite using markers for postsynaptic density, and then you can determine GR phosphorylation dependent effects. So that's the question that we ask, whether GR phosphorylation has any role in dendritic spine number. So the answer is yes. So elegant work by Freddie shows that when you culture neurons for several weeks and you treat them with BDNF and dexamethasone treatment, Freddie was able to use an shRNA to GR to knock down GR expression, And you also are able to reduce the number of spines on a dendrite using this SHGR. Now, you can rescue that phenotype by complementing with a construct that's resistant to the SH endogenous GR with a GR wild-type construct. Now, if you use a phosphorylation mutant, it mimics the effect that you see with the GR knockdown, suggesting that GR phosphorylation plays a role in mediating dendritic spine number from GR upon BDNF plus dex treatment. So we went back to our arrays, and there was one particular gene, NGFIB, that seemed to potentially have a role in mediating dendritic spine morphology and number. And I've shown you before that this gene is sensitive to GR phosphorylation, since the double mutant diminishes activity upon co-treatment. And then by Western blot, you also see reduced levels of the protein, while GR levels generally stay the same. So the question that we asked, does NGFIB have any role in mediating dendritic spine number? So if you take mice and you chronically stress them, or you inject corticosterone, which is a type of glucocorticoid, you'll find that spine numbers are reduced compared to wild type. Now, if you use, Freddie did an excellent experiment where you can electroporate shRNA for NGFIB into the in utero of the mice. You'll find that this effect is abrogated, suggesting that NGFIB is mediating the downregulation of dendritic spines by stress and chronic glucocorticoid treatment. This effect is also dependent upon GR, since using a TET-on system where in both black and white bars, NGFIB expression is induced. You can have the white bars that after 10 days would lead to decreased spine number, and the black bars which have SHGR and knockdown of GR expression, this effect is abrogated, suggesting that it's dependent upon GR expression. So in conclusion, NGFIB regulates dendritic spines in a GR-dependent manner. And to summarize, we find that GR phosphorylation at both serine 155 and serine 287 contributes to BDNF-dependent induction of select GR target genes, and that GR phosphoisoform recruitment is not selective to SGK1 or GILs. But in another post-recruitment effect, GR phosphorylation appears to alter the recruitment of a coactivator, P300, and a transcription factor, at least at SGK1. And we think that CREB is required for BDNF-dependent GR-mediated activation of SGK1, but not for GILs, not for that insensitive GR target gene, phosphorylation insensitive. And GR phosphorylation in serine 155 and serine 287 appear to facilitate GR induction of NGFIB to modulate dendritic spines and synaptic plasticity. And we think that this may have implications for glucocorticoid and BDNF crosstalk in learning and memory, or those diseases and disorders that may be affected by dendritic spine number or morphology. So there are a few future questions that I would be interested in addressing that correlate with this project. One is CREB implicated in GR-dependent induction of neuronal genes. So I showed you data from SGK1 knocking down CREB, and it seems to have effects. You could also look at our neuronal genes that we identified in the microarray to see if CREB also affects the expression of those genes. It would also be interesting to investigate other transcription factors that may mediate crosstalk with GR that are downstream of BDNF and TrkB signaling. We went back to our arrays and we found that using motif analysis, a number of transcription factor binding sites were enriched in the promoters of BDNF and dexamethasone-induced genes. So of course we found CREB, but the other transcription factors like SP1, which has been shown to interact with GR in CVP and KLF7. So it'll be interesting to knock down these factors to see if they have an effect. The other question I'd like to ask, does BDNF affect GR recruitment? to DNA genome-wide. I showed you data where BDNF treatment can enhance GR recruitment to the promoter. What effect does this have genome-wide? Maybe it's possible that BDNF is able to enhance GR recruitment to new sites on the genome. So I think a GR chip-seq experiment will help us to kind of understand that. And then finally, I believe that there's some type of relationship between these composite GREs, or GREs that contain another transcription factor binding site that contributes to the effect of GR, and phosphorylation sensitive genes. So the question we would ask, are genes that contain these composite GREs more phosphorylation sensitive than genes with simple or multiple high affinity GREs? So we could examine DEX responsive genes for GREs and CREB responsive elements within the promoters of the phosphorylation sensitive genes. And then even a CREB chip-seq experiment using our stable wild-type GR expressing cells or the double mutant expressing cells, we would hope to see differential effects of CREB recruitment in those two cell lines. So that pretty much wraps up my talk. I just want to really acknowledge the people who helped contribute to this work, or at least helped me in my graduate school career. Definitely Michael Garabedian, I'm really indebted to allow me the opportunity to work in his lab. And I've just really enjoyed the time working with him. I think he was just an excellent PI for me. Claudio, Herb Samuels, and Naoko, thank you very much for contributing to my thesis committee. And then Inez for taking the time to come over as an outside reader. I'd also like to thank Joel Oppenheim for your help and helping to make my transition into grad school much easier. We'd like to acknowledge our NIH grant. And then Moses Chow and the Chow Lab, Freddie Juntu, who performed much of the work that you saw here. He's been a tremendous help for my graduate school career. I definitely could not have made it to this point without his help and just those conversations and critiques. Thomas Newber for the mass spec and Chongfei Zhu for the mass spec experiments. The Chow Lab, thank you very much for your cooperation and advice and support. The Garabedian Lab, you already know. The Susan Logan Lab, thank you very much as well for your help over time. And then I'd like to also thank my family, my mother and stepfather who are coming later in the week. And then I have two children. My son Amir is 3, and then my daughter Anissa, she's 6 months now. And then my sister and the Basir family, thank you for your contributions. And then last but not least, my wife, who's I think deserves all of the dedication and support, because this was definitely not possible with 2 kids without her support. And thank you very much, and I love you very much. Thank you.
Marcus will be happy to take questions from anybody in the audience except the committee members.
Yes. I have one small. It was a nice presentation.
Thank you.
Have you used any BDNF knockout mice to see the glucocorticoid signaling?
So we've used transgenic animals that have reduced BDNF expression, like the BDNF hets and the TrkB hets, and we've assessed GR phosphorylation, and we noticed that there's reduced phosphorylation, but actually gene expression or glucocorticoid receptor signaling, we haven't particularly assessed in those animals. Yes?
Marcus, you mentioned that overexpression of GR caused resistance to stress. Is it known how that works?
It's primarily through not being able, actually being able to better repress the HPA axis and hyperactivity of the HPA axis upon stress. I didn't really talk about the HPA axis, but it mediates, it's basically a direct axis that mediates glucocorticoid induction upon stress, and GR is able to have negative feedback on that pathway. So in the GR overexpressing mice, they think that HPA axis is able to be better controlled.
But that's phosphorylation dependent?
We're not sure. We haven't tested, we've tested CRH, but not some of the other POMC, which is important Yes.
Do you know if GR activity is regulated in inflammatory states?
Yeah, most definitely. I haven't, looked at inflammatory states, but I'm even sure that there may be some other, crosstalk between other signaling pathways in GR that may, help to modulate GR in inflammatory cells as well. Yes.
Are there any behaviors or anything about being BDNF deficient mice that might speculate involve glucocorticoid defect in the crosstalk between the two?
So in the BDNF hex and overexpressing BDNF in mice is not much of a phenotype, but there is a Val 66 met mouse which is basically a polymorphism SNP inside the BDNF gene that misregulates BDNF translocation to the projections like the dendrites and axons. So you can have BDNF production, but it's not reaching the areas of the neuron where it may be important. In that mouse, actually, you see phenotypes related to stress and having HPA axis misregulation. But that can be compensated by antidepressant treatment.
If there are no other questions, I want to thank Marcus, and also whether Ranger shines, we'll have a little get-together afterwards in the third floor Squareball Conference Room around 3:30, so you're all welcome to come. Thank you.